late-stage ovarian cancer. And this isn't working, so I'll give you a...
Okay. Thank you everyone for coming, and you're either not soccer fans or really excited about what I have to say, so I'll assume that it's the latter. Thank you very much for foregoing the World Cup to be here. This, by the way, is a highlights track presentation and a paper that has just recently come out in Journal of the National Cancer Institute. So this actually does have a lot in common with the talk that you just saw, but just for a different objective, and just to very briefly tell you what it is about. I am interested in prediction and looking at the literature of studies that have taken microarray data and used that to try and predict outcome of cancer. So there's a whole arsenal of tools that people use to do this. The general approach is the cross-validation approach, like you see here, where you split one data set up and iterate like this and use any number of different methods, or maybe a bunch of them, and choose the one that works the best, and then go ahead and test it in some left-out test set, potentially using publicly available data. So I'm interested in ovarian cancer, and this has been a very common, it's not a super common cancer, but it's one where there have been a lot of efforts to do this, even relative to other more common cancers. And I think part of the reason for this is that although it's not the most common cancer, it is one of the top killers. It's a deadly disease, and it has not kept pace with other cancers in terms of a 5-year survival improvement over the past 5 years. And importantly for this study, most of the cases are diagnosed with late-stage, high-grade serous disease. So unlike studies, say, in lung cancer or prostate cancer, where you have very good clinical markers, we don't have that in ovarian cancer. There's actually very little to go on for most patients in terms of predicting prognosis. So there have been lots and lots of efforts to predict prognosis for late-stage, high-grade serous ovarian cancer. And I focused on a 5-year period in the literature by systematic review. These are actually the papers that I studied for this meta-analysis. The objectives: first one was to assess the reproducibility of published models, and this was happening just in the wake of some fairly big scandals in the field at Duke, so this was really of interest to us to see whether this was representative or an outlier in the field. And then to take all of these studies and evaluate them using publicly available data. And of course, having collected all of these data, it's natural we'll want to see if we can use it to do a little bit better ourselves. And then finally, to take the most promising models and validate them again on formalin-fixed samples from a clinical trial. This meta-analysis, I can describe it in 2 main arms. The one arm here has to do with the data. So this involved going to Gene Expression Omnibus, authors' websites, everywhere, to collect all of the data available and put them together into this curated ovarian data package, which is in Bioconductor now. There are about 24 studies from there, of which, so I set a number of inclusion criteria around sample size, around the comparability of the disease, that they are late-stage, high-grade serous, and primary tumors, et cetera. Filtered that down to 10 data sets and about 1,250 samples. The other arm of this is the models themselves. These are the published models that people have claimed have prognostic significance for ovarian cancer. This was a systematic review with a defined PubMed search, turning up about 100 articles over a 5-year period, and looking at those in review papers, having a set of defined inclusion and exclusion criteria that was whittled down, and finally being able to reproduce the model was whittled down to 14 prediction models for overall survival. All of these efforts are detailed in a very large supplement to the paper, so you can actually see what was done to assess and reproduce these models. So as that first objective, reproducibility, it was actually not bad. And I think a lot of us had the impression that it was rampant, that nobody's work was reproducible in this literature. It was not really the case. There are some that were definitely not, but most of them were. So there are these columns here. The first one was kind of the most important from my perspective. Did they provide a fully specified model? And this is something now if I'm reviewing a paper is like first thing up front. If you're proposing a model, are you actually saying what it is so that somebody else can take it and apply it to different patients? And that requires putting a fair bit of detail into your supplement probably so that somebody else can just go and reproduce your model without having to go through all of the steps that you did. But if it wasn't, it still might be possible to reproduce the model using the training data, using to follow the steps and try and recreate the model. Then another important part of this was taking test data that was used in the paper and seeing if we could reproduce their validation of the results. And then if we could, then that was kind of this final column. It's verified. We know that this is a good representation of the model. So the main result, you could say, of this is the actual assessment of the 14 models in these 10 data sets, and this represents all of those assessments. So there are the rows here, 14 models. Oh, can you see that? Yeah. 14 models in the rows, 10 data sets in the columns. In each one, I'm calculating a C-statistic, which is just like an equivalent of area under the curve for survival analysis. It's 0.5 if you have a random predictor, and 1 if you have a theoretically perfect predictor. You'll notice here that, so it's color-coded. The black boxes here are training sets, so that's where we expect there to be overfitting. These are not used in the assessment. It's a bit hard to see, but there are some gray boxes. Those are test sets, which should not influence the model, but give us the opportunity to test for publication bias by comparing those to other different data sets. So just to give some context here, one model and one data set. This is something that if that's all we were looking at, you might represent as a Kaplan-Meier plot. And one row would be a meta-analysis. So this is really 14 meta-analyses, which is why I called it a comparative meta-analysis. So each of those meta-analyses can be synthesized, and we get a point estimate and a confidence interval for each of these models. The best model here is the one produced by the Cancer Genome Atlas, which had the biggest data set. They had good bioinformaticians and statisticians. They used good methods, so maybe not surprising, but what was more surprising perhaps is there's a whole group of studies in the top half here, which actually have a lot in common with each other. And when I looked at the genes used in the model, there's almost no overlap. That's not surprising. When I looked at pathway enrichment, functional enrichment of those genes, again, basically nothing in common with them. But if you take those predictions, you see that the predictions are actually very similar. And if you look at the genes that are correlated with those predictions, those have a lot in common. So these top 5 models here, they're highlighted in yellow. Doing a gene set analysis this way by looking at the actual predictions, they have very similar results, and they're all associated strongly with these immune type pathways, and specifically a high-risk score is associated with low expression of these immune response pathways. And interestingly, I thought none of these papers interpreted their models that way. So there are a bunch of findings from this. I'm just going to highlight one more here, which is actually looking not at the models, but at the data sets. So one of these data sets was a controversial one. It came from Duke University by Dressman et al. That was criticized for having a run batch effect that was confounded with outcome. And it was eventually later retracted, although for other reasons. And by comparing this against all of the other data sets, we could see the average validation statistic here is 0.61 compared to 57 for the next highest one, which is actually takes a bit of calibration to get used to this C statistics scale, but that's a big difference. So it means that any model that you test in that data set is going to look pretty good. And interestingly, when I did an assessment of the magnitude of the batch effect, so this is a histogram of associations between each gene and the run batches. And so if you see a wide tail like this one on the top left, that means there's a very strong batch effect. Lots of genes are associated with batch. If you look at this Tothel data set or the Dressman, it's not an especially large batch effect. So the lesson here is that you can and you should do everything you can to reduce batch effects, but you cannot eliminate it. And if it is associated with outcome, then you're going to be out of luck. There's nothing that you can do with it at that point, even if it is a small, well-controlled batch effect. So that's all of the results from the comparative meta-analysis that I'm going to show you, and I'm going to talk a bit about some other work now, including this was a companion paper that came out in the same issue of Journal of the National Cancer Institute, which was taking all of these data sets to try to develop better predictive models. So the most exciting result we had from this was actually for debulking. So this standard treatment involves a removal of as much of the tumor as possible, but in about half of the cases it just turns out to be impossible. And it's thought that in these cases those patients do not benefit from the surgery. They go through this very invasive, dangerous surgery and get no benefit from it. So if they could be predicted in advance from a biopsy so that we could know that they're not going to benefit or are very unlikely to benefit, they could be triaged and taken instead to an adjuvant therapy with interval debulking after the tumor has been shrunk. This is currently in practice, this adjuvant therapy approach, and it's been shown to be equivalent to the standard surgical debulking. So there's no benefit or harm, but if these patients could be stratified better, there may be an advantage. So how do you use a bunch of data sets, both to build a better model and to assess it? The approach we took is very simple one, just doing univariate regressions in each data set separately. So each of those 10 data sets, or there are a little fewer here, use a meta-analysis to synthesize the coefficients. So these are fold changes, or in the case of survival analysis, it's a Cox regression coefficient. And then once you have that synthesized coefficient, you just average gene expressions in any test set using the weights using those coefficients as weights. So this is what I call a univariate ensemble method. It's about the simplest approach that you could take. And then we took what we call the leave one data set out cross-validation approach, where I'll just show that to you on the next page. You would take one data set, don't use it, use all of the rest to build your model, and then test it on that one that you held out, and then continue and repeat this one by one. So what you'll see here is some of the data sets, like this top left one, doesn't work at all. In most of them, though, it does work, even though it is fairly modest. So based on the microarray data, we were quite confident that there was a true biological signal here, that this was a predictable thing, but we were just not sure that it could be predicted accurately enough to be clinically useful. But we had a collaborator who then took this to a tissue microarray, picked out 3 proteins from his favorite pathway, and had a few pathologists score this in several hundred tumors, and it was just striking how much better, how much more predictive this was. I think both from the fact it was done at the protein level where the function is, and that it was done in a consistently well-annotated set of tumors. So there's just taking these 3 proteins and adding their expression, we're up to an AUC of 0.9, and just looking at the top, hit, this periostin gene, there is a huge difference in the surgery success between non-expressing and overexpressing of periostin. Okay, I just have a sort of one slide each of a couple other related projects here. We've been doing a lot of work on using a collection of data sets such as this for validation in comparing prognostic models, also in comparing algorithms. And the approach that we've been promoting and developing is one where rather than just looking at cross-validation in each data set, we look at all of the pairwise combinations of training and test sets and build them into a matrix like this. And there was a talk this morning by Christoph Bernau, which is in the proceedings track, talking about the basic what are the properties of this matrix in simulation and in real data, and how does it compare to cross-validation. And there's a second paper building a Bayesian clustering method to use this to identify outlier data sets. I think it's very interesting because the things that impact on your outcome do not necessarily impact on prediction models. If it's something that is captured in the gene expression, then you can have, a difference in something, in some known clinical prognostic factor that influences outcome, but it's okay if that varies between data sets. And then there are other things that might not directly influence outcome, but actually might impact on your ability to predict using gene expression. And we're calling this the Leave One data set in matrix of validation statistics. Another thing that I'm working on currently is automated methods for identifying duplicates in these large databases. So for ovarian cancer, 3,000 samples, it turns out that about 17% of them are non-unique. So they have a duplicate somewhere in the database. And just cleaning them all up caused a loss of about 10% of the samples in the database. These occur any number of different ways. The most common one is one institution keeps publishing papers with the same samples, but they might even use different technologies. They add more patients to it. It also occurs with sharing between institutions. You can have 2 institutions across the world from each other, but they have duplicate samples because they've both been collaborating with the same hospital. And it occasionally occurs within a single data set, although that's more rare and more of an error. Somewhat disturbingly, about a quarter of these had discordant clinical data, so inconsistent with each other. In one case, the patient survived 5 years, in the other case, they died in 3 years or something like that. So here are the first 2 manuscripts I told you about already. These couple of other ones are ones coming out soon in bioinformatics. And this was just investigating using cross-study validation, a number of different algorithms, but comparing them to this one very, very simple algorithm. It's about as simple as you could possibly have and have something that still worked. And we found that it is competitive with much more popular and sophisticated algorithms. So I have some sort of bioinformatics centric conclusions for the ovarian cancer. There are lots relating to ovarian cancer, but I think in terms of what it means for you, sample size, I think, is really key. This is, I've seen in several cancers now, more so than the actual algorithms that you use. Publicly available data is like we saw in the last talk, and like I have seen, a wonderful resource, both for building more robust prediction models, for evaluating claims that have already been made in the literature, and also for identifying potential problems that have happened in individual studies. None of the models that I evaluated were sufficiently accurate to be clinically relevant, but I think it's worth emphasizing that they were better than random, and I think that additional investigations, especially at protein level, are definitely warranted. There are a few curated data packages now for doing studies like this for ovarian cancer, bladder, and CRC, and I expect there to be more on the way as well. All of these are reproducible. You can look at the paper and it'll give you a link to the Bitbucket or other web page for reproducing this. One more thing that I just wanted to mention, I will be involved in more integrative analysis, but looking at multiple genomic assays such as the TCGA. This is a project that will be starting up in the fall with colleagues from Fred Hutchinson, Johns Hopkins, and Harvard, and we will be looking for programmers and postdoc positions. So please let me know if you plan to be looking sometime in that timeline. So thanks very much, and take any questions. Yes?
Yeah, hi. Often when we're trying to apply one study to another in exactly the way you've done, we find that key relatively what appears trivial information regarding normalization is left out, which makes it basically impossible to apply one model to another. And I wondered if that was your case.
The only studies that I saw where it was really impossible to apply models across were where it was like a 2-color microarray or something really different between like Agilent and Affymetrix and different Affy platforms. It didn't seem to be a big difference for me in these studies, and also in breast cancer. It was things a bit more, either more subtle relating to the patients or just some really different platform or really different normalization method, like one where all of the columns had been standardized to a z-score. That made a big difference. So I have, I guess, a more optimistic look at that. It seems that the platforms and normalization, I mean, I did a lot to make those studies as comparable as possible, reprocessing them all from raw data where possible. But I think it is possible. And in the doppelganger project, I found that it's possible to identify duplicates even from very different platforms, even from RNA sequencing as well.
So I'm going to continue that question.
Yeah.
In terms of the linear model that you presented, are there mixing coefficients for the different models? So then that would account basically for different types of normalizations, because you can expect genes in RNA-seq, however you normalize them, versus gene expression in arrays to have completely different values, in which case you can't just linearly combine them.
Yeah, I think that for one thing, the successful signatures in ovarian cancer are all quite a few genes. Like, you tend to have a lot of genes with very weak associations, and you do better by averaging a lot of them together. So I think that in a way it kind of averages out a lot of the platform differences. The only like absolute standardization that I did for these was to give them the same mean and variance in each platform. But
so another question, you're saying that all these studies had different lists of genes that came out, and then they did not intersect, but they were enriched for a similar category.
yeah.
So my question is, if it's L1 regularization, or why isn't there an intersection between those different studies? There should be some type of intersection.
Yeah, there's at most 2% intersection of the genes. I think the reason that there is not much intersection is that you have a highly correlated transcriptome. And when you do an association study with your outcome and rank your top genes either through regularization or filtering, it is even with comparable studies repeating the same thing, it's going to be quite random which genes appear at the top. But because they're correlated with the other genes, you're still getting the same predictions. So like that case where you have only 1 or 2% overlap, you can still have a correlation between the predictions of 0.6. So looking at the predictions, they're the same model, they're just using different genes to do it.
But wouldn't L2 regularization correct for that? So then you'll get groups of correlated genes having similar weights.
Well, I guess if you were inclusive enough and you had like thousands of genes in your model, then you would have quite a bit of overlap. But if you are going down to 200 or fewer genes like most of these do, then there's not going to be much overlap.
Okay, thanks.
Sure.
Hi.
Okay, one more question. Yeah.
So you mentioned that the 3-protein simple model outperformed the mRNA model a lot better. So have you checked just the mRNA level of those 3 proteins?
Yeah.
Do they
Yeah.
work?
No, they don't work like the proteins do.
Do they work better than the more complicated?
No.
Not really. Okay.
No, they don't work as well either as the more complicated. I think that at the RNA level, the averaging effect of these noisy measurements, averaging a lot of genes is important. But yeah, at the protein level, it just having those, even just one protein was more accurate than its corresponding gene or all of the genes, and that one gene is less accurate than using all of the genes as well.
Okay. Thanks.